Thank you very much for joining us today for the finals in our series of credibility lunchtime seminars on neuroimaging. Today's speaker will be talking to us about research data management, which is an incredibly important but sometimes underappreciated topic in neuroimaging. So I'm very pleased that we get to spread the important news with you today. So my name's Cassandra, my pronouns are she, her. I'm the Open Science Community Engagement Coordinator for the Wellcome Centre of Imaging at the University of Oxford. Our speaker today, Etienne, will be speaking for about 15 minutes, and then we'll have the rest of the time for Q&A. So the Q&A box will be open throughout the seminar, and if you want to just pop your questions in there, and then we'll put them to Etienne at the end. As I said, this is the last in the series, but the two previous sessions are recorded and available on the BNA Credibility website and also on YouTube. So please do go back and have a look at those if you haven't watched them already. So then, just to introduce Etienne, he's the Associate Professor in the School of Psychology and Clinical Languages at the University of Reading. He's also works very closely with UK Reproducibility Network, and he's also got a lot of informatics experience, which he will touch on in this presentation. So Etienne is very well positioned to talk about this in terms of reproducibility, but also the kind of computational side of things. Have a look at the chat for information from our organisers. BNA and Verena is also putting things in the chat for, use that for general discussion between yourselves and any important messages that you want to share with the group. With that said, I'll hand over to Etienne now, and I look forward to joining you all for the discussion again at the end. Thank you.
Cool. I think I'm sharing my slide correctly. Is that right?
Yes, it looks great. Thank you.
Awesome. Thanks. So thank you very much for inviting me. I'm also very happy and grateful to the organizers to have moved the talk from the strike period. As you know, the UK institutions are on strikes for good reasons. I do work in the Center for Integrative Neuroscience and Neurodynamics at the University of Reading, and I do a lot of work on reproducibility, both at Reading and also through the UK Reproducibility Network. Just to give you a bit of context, I started as a software engineer. I literally got bored of writing code every day and wanted to understand psychology. So I studied psychology, and now I'm in a lucky position to be able to do both at the same time. I'm mostly an applied statistician these days. I develop methods and I teach statistics and programming. So it'll give you a bit of context of where I'm coming from in the rest of the talk. If you are really tired, you can fall asleep, but this is ideally after this slide. So I'm assuming that you're all familiar with the reproducibility crisis. If you're not familiar, I strongly encourage you to read these papers. In my talk, I want to convince you that credible research is just research done well. Unfortunately, there is no app for that. So I know that my talk is about bids and FAIR, and I will definitely speak about various apps and things that you can use. But I really encourage you to think beyond the app and take a longer perspective. I imagine that we can speak about that in the questions as well. So what do I mean by credible research? I really like this definition of research, which for posterity, I will read it aloud. So for knowledge to benefit research and society, it must be trustworthy. Trustworthy research is robust, rigorous, and transparent at all stages of design, execution, and reporting. And what that means to me is it gives me a structure to think about how I should think about my agency in doing research, basically, and my responsibility as a researcher. And the way that I teach that, so these are literally the three first slides that the students in the masters that I'm teaching on see. I can be absent-minded on Monday morning at 8 or 9, and so I do put a slide for myself to remember that I need to take attendance. But that's also the very first lesson and sort of the core lesson that I want the students that I teach to remember. So I teach statistics for fMRI and reproducible analysis. And this is a core thing that I want them to remember, is that in order to do reproducible research and credible research, a good scaffold to think about it is to be selfish about it, to imagine maybe in 6 months, a year, 2 years, I want to go back to my studies, to my research, to my results. What do I need in order to make my life better? It's the same way as the same way when you want to go to the gym and you put your gym bag next to the door so that you don't forget it. It's the same principle. So the way that we keep notes about data has evolved. These are the famous notebooks from William Gossett, who you may or may not know is the inventor of the student's t-test. And the reason it's called the student t-test is because his notebooks where sort of the marketing said the student science notebook. And also he was working for the Guinness Brewery at the time, and he was not allowed to put his name on the publication. So his pen name is student, and this is why it kept like that. From the notebooks, we have evolved to using bids and other various tools, and I'm going to sort of describe the landscape of the tools that we have at our disposal in neuroscience generally, and where I'm hoping things are going. So BIDS stands for the Brain Imaging Data Structure. Although it started because there was a platform called OpenFMRI, which I'll speak about in a minute, that was designed to be a sort of a centralized place for people to analyze their data. And they had the simple problem that they had to manage a lot of data in a very consistent way on one server. And so how do they do that? And so they derived this way of organizing the files in a very reproducible, very robust way that later became BIDS. Now BIDS is actually a very big project. Just the specification document is 500 pages long, because there are so many use cases that one would want to think about that it can become very messy, become unuseful, or difficult to use, and so you would have to think about the development. Actually, now I do want to put a disclaimer a little bit. I should have put a disclaimer a little bit earlier. I'm not involved in the development of any of the tools that I'm about to describe, or in BIDS. So there might be some mistakes or omissions or that in what I describe. But at the same time, I think it's a good thing that I'm not involved, because I take the sort of plain user approach view on these things, and so I do have a bit of a critical view on these tools. BIDS is very simply a recommendation on how to structure the folders hosting your files and your data. On the left-hand side, you see, I hope that you see my mouse cursor here. On the left-hand side, you see the recommended architecture and the different types of files that you can be hosting here. In this case, this is BIDS for EEG, because it started with MRI, but it has evolved in many other, to include other modalities. And the idea behind BIDS is really to make it easy and for a human to read and understand what is going on. And so these are all text files with a very specific structure that is also flexible. And so it's a very nice way to organize your data, basically. I've put a number of links in my slides. You can find them on the slides when if you want to copy paste them. So even if you have the recommendation of the specification, it might be a bit difficult to organize the slides because you have to do it manually, or you can do it manually if you want to. There are different tools to help you to organize your data with BIDS. Now on the slides you can see how the BIDS validator work. So it's a web-based interface that will just read the structure of your folders, and the subfolders will read the text files and try to figure out if there are any mistakes, and it'll flag out and validate, assert if the if your structure of files is appropriate or not. It doesn't correct any mistakes, unfortunately, but it can be a bit picky on some things. So but it works pretty well. So it actually helps a lot. If you do not want to do it necessarily manually, there are tools that help you to convert your folders into a BIDS format, depending on what you want, the modality, depending on the software that you are used to using. And on the BIDS website, there are quite a few lists. I was actually really surprised to see that there were so many converters, as they call them. So for MRI, for example, there's 20 different converters that exist. Some of these converters are actually using they are children of each other. So they're using, and they have some dependencies on the converters. So it's a bit, it can be a bit tricky. One thing that I want to point out is that from like many of these projects that I'm describing, these are community efforts that, as you know, with technology these days, sometimes it breaks, or anyone who has received funding for a project knows that at some point the funding ends, and when there is no more funding, there is no more development. And so this is, in my view, one of the biggest challenges that we'll face in coming years is how do we maintain these tools, and I think that's going to be a bit of a tricky issues. Once you're able to organize the data appropriately, one nice thing that you can do is to store them and share them. And so Neurovault is based on BIDS. It allows us to anyone to share data in a very easy way. I do recommend using it because it's a nice it's sort of curated by the community, so it's a nice place to go to share data. I wasn't able to find information about the funding, however, and I have experience managing the data from the 140 users in my center. And I know that we have 60 terabytes of data, which is a lot, and that costs a lot of money. So I at the moment, it is paid by grants. So Neurovault is paid by grants. When the grants run out, what is going to happen? I'm not quite sure. So do not just use that as a backup, or put your data in there without any backup, because it might disappear. Once you have BIDS, the nice thing about having a robust and consistent structure is that you can literally devise many different tools. You can share processes, and so Repronym is a nice community. It's North American. They developed a whole suite of tools, and so I just copy pasted the list of the tools that they have available. One hugely useful tool I find is DataLab, which allows you to query Neurovault and query different databases that exist, and you can actually look at the data without having to download it necessarily. I guess the difficulty is that you do have to be what is the word? Able to manage the command lines, and so it can be a bit tricky to get into this, but they did put a lot of effort into the DataLab handbook to explain how to do this, how to do these things. Once you do have an ability to store data, you might also want to share analysis platforms. And so I just picked up two here. I know of more that allow you to query and redo analytical pipelines on the cloud, which are really useful. And all of this is provided because we are able to create a robust structure to store the data. And once we've done that, we can even go one step further and we can combine analysis and run meta-analysis. And so these are two of the platforms that I know of. The Neuroscience, I'm not quite sure what the state of development is. I know that Tal, the main developer, has left academia and went to work for Twitter. And then I think, I'm not quite sure he's even working for Twitter anymore. But so I'm not even sure that Neuroscience is still under active development. So that's part of I guess, the general issue that I see with using these tools. So now let's assume that we have a reliable way to store the data and we can describe it. How do we turn this data into knowledge generally? The BIDS is really made for human readability. It does use text files and JSON formats that allows computers to do things, but to read the files and do some things. But there is a gap in wanting to combine all these data into a bigger pool of knowledge. The way I like to think about this is that in 50 years, how will our children or grandchildren manage to make sense out of the data? And at the moment, I'm not quite sure that BIDS is a solution, for example. And so one, I guess BIDS leads to FAIR. FAIR is a set of principles that anyone can apply to managing their own data. And it can be, the way I see it is that it's a continuum. Data should be findable, accessible, interoperable, and reusable. And so how you do that depends on your use case, depends on the discipline, depends on the quantity of data you have. Obviously, if you have text files, it's a bit easier than if you have DICOM files. And so the way to describe these, there is a bit of a gap in just being able to store data in BIDS and then make it, turn it into a FAIR format that allows further exploration in 50 years. FAIR is actually, it sounds nice, and it is nice, but making it work can be also a bit tricky. So this is a nice description on how to turn your data into a FAIR format. You obviously, once you have the data, you have to define a semantic model to actually understand what the data explains and how to best store it, because you cannot just store it in a Dropbox folder and say, well, this is linkable, done. You have to make sure that the people who can retrieve the data actually know what they're retrieving, and that is where I think the gap still lies. So this is a very nice, I think it's called the Semantic Webs cake which represents the concept of information as it goes from the hardware to the representation of knowledge and trustworthy. And so you can see that the latest part of the cake is a trust that we have in the data and the knowledge that we produce. And I think that the bits, for example, so it goes from the lower level protocols at the very bottom to the trustworthy knowledge that we have at the very top. And I think BIDS is somewhere between the first and the second layer. It's not even using RDF, which is a very common format that manages many aspects of the web and the online resource we're using. And so I think that there is a bit of work that needs to be done in order to turn our neuroscience data into something that we can actually reliably use and trust as we go along. I want to leave you with two quotes that I really liked as I was researching for this talk. These are from the people who invented the web a long time ago. Tim Berners-Lee, who invented the World Wide Web and the technology behind clicking links and creating the World Wide Web and the internet as we know it. The web of human readable documents is being merged with a web of machine understandable data. And I think this is where this is going. You all have used ChatGPT and you are all probably very amazed by how it manages to do things, but you have noticed that it's only really good at describing the training data set that it has been used. And if you have asked ChatGPT to convert a piece of code to another language, or even speak from English to French, for example, you will see that it's not being very useful. And so the only way for that to happen is to actually add this layer of knowledge on the data, which I think is still lacking. So that's it. I am hoping that I have convinced you that credible research is research done well. I'm hoping that that I haven't really turned you off from the various apps that exist, but I do urge a bit of caution in using them. It's not because you're using BIDS that everything is reproducibility and credible research done. I think that there is a bit of thinking that needs to happen in a bit of a more forward-thinking process. And that is it. I'm happy to take your questions.
That's lovely. Thank you so much, Etienne. So we've got one question in the Q&A and I've got a few myself. So do feel free to keep your questions coming in the Q&A, and we'll probably end just a little bit after 12:30. But of course, anyone that needs to leave at 12:30 on time, you're very welcome to jump off when you need to. So thank you again, Etienne. A couple of things I found really interesting there is that we talk about RDM and like informatics as this kind of essential part of the project or the process. And particularly for people that are coming from perhaps more like clinical perspectives, this feels like the hard bit of the work, right? Using the terminal, like you said, can be a big barrier for some people. But I think it's really nice, and I haven't seen people talk about it, that there's the top layer, the kind of the abstraction and the interpretation, and that the layer of trust and the knowledge part of it, and all of that is made not necessarily possible, but easier by having a coherent, robust structure to the data underneath, right? Because your abstraction and your knowledge generation can go completely awry if you haven't got a very strong foundation underneath. So thank you for bringing that to our attention there. So we've got one in the chat, and then if no others come in, I'll put all mine in there as well. So someone has asked, what's the difference between Neurovault and Open Neuro? And I also just wanted to flag that we had a talk from Cyril Panay last time, and we know that Open Neuro isn't appropriate for data generated in the EU. for GDPR reasons. So
yeah.
So what is the difference between Neurovault and Open Neuro?
And actually, Cyril, I think, is now on the steering group for bids, if I remember correctly. So so Neurovault is an online cloud-based repository of data, and Open Neuro is an extension of bids. It's a bids apps that allows you to analyze data sets, and so you can share analysis as well. It allows, yeah, it's basically an analytical pipeline. There are a few others that exist. Biscuit is or Boutique as well, and there's how is it called? Seabrain. It's a Canadian version of that I know of.
And so the way I think about the two different, like in terms of repository, is not so much of the analytic platforms, if this is useful for folks, is that Neurovault, I would put my maps, like my Z or T maps, like probably Z, and if I was able to use Neurovault for Open Neuro for data, you put more of the raw data in.
That's right.
So so Neurovault is good for when you can't share the raw data or the raw form, you can still share the analyzed data.
Yeah, and as you mentioned, because most of these, pretty much actually all of the tools that I spoke about today are hosted in the US. I don't know if they are equivalent in Europe, but
not yet.
Not yet.
Yeah, we'll get there. So Verena's put a question in, which is kind of, it's not it's not RDM specifically, but you touched on it, so it's worth mentioning that a lot of these tools are developed by communities, and what happens when funding runs out. So what are your thoughts around sustainability of these things and sustainability of the research products that we create and rely on so heavily in general?
Yeah, so unfortunately, I don't have a solution, and I don't think that we can get away from the community-led efforts. I think that at the moment, this is the most credible way of doing things. Having said that, it is true that when funding runs out, then the developers have to find a way of paying their rent. And so then the tools become obsolete or deprecated. There are bugs happening. And these days, the turnover of libraries and dependencies and security risks, it's by the week, and it's no longer by the year. It's really crazy. So I think the best recommendation I would have for us as a community is to become better at coding and understanding the requirements for these things, and not to jump on the next Twitter-fueled craze for a platform that looks nice because they have a nice marketing person, and then it just dies out like that. And so I think that there is a bit of work for us to be mindful of this and sort of think long-term, basically.
Okay.
I guess that's the main message of what I wanted to say in the talk.
And then so going back to the RDM side of things, I had to do a little calculation about when I started my PhD. So it was about 15 years ago. And
I can tell you that.
Yeah. So I remember in my kind of second year, I devised my own system for structuring my data, which I was very happy with for a few months. And then I realized that was complicating things. I was basically putting like the date of the acquisition in my file name. So then every time I had to call that and I had to loop through these things, I had to specify the date, and it was getting so I reorganized everything. And I think I did that about at least twice, maybe 3 times. So I very much feel the pain of having to make your own data structure like solution. And so it was easy for me to recognize when bids came around how valuable it would be. And the kind of the community input, you've got all the experience of all these different experts developing something. And then as a result of this sort of standardized structure, all these different tools could be developed around it, and I can just plug my data in. So having felt that pain, it was obvious to me that this was a great solution. How do your students respond to it? And do they just take your word for it that this is a good structure? Or is there some like exercise that people should be doing to go, oh yes, it is rubbish when I develop it by myself, and I should look to the other ways of structuring it?
This is a really good question. I'm very positive and optimistic about this. From the time that you and I were doing our PhDs, things have changed quite a bit. And when I describe these tools and these processes, the students typically wonder, is this not how it happens? How did you do it before? That said, most of the students I meet are from psychology or neuroscience background. And there is a big hurdle of having to open a terminal and then understanding the ins and outs. And as you know, I don't necessarily make it easy on my students by teaching them a GUI. I do think that the GUIs create a bit of distance between what you do to your data and what you understand is happening. And the shorter that distance, the better. Even though the students find it very appropriate to have these tools and so on, they do all have to pass the hurdle of having to learn how to do these things. But actually, as I said, I'm very optimistic. The students really take it very well. So.
Which reminds me, when I think about other communities, other disciplines, and I have the same reaction, like, you don't have a standard. You don't have a standard. So I think neuroimaging really is quite far ahead in some respects compared to other disciplines that we're tackling these issues, and that there is such an active community. I know folks, our two main sort of camps of OHBM and ISMRM, there's really active reproducibility communities in both areas. So I'm very surprised now when people haven't encountered these kind of topics when they come to us as new students.
Yeah.
Very positive. Lovely, thank you so much. So we are just slightly over time, which is absolutely fine. So thank you, Etienne, again for sharing your experience and knowledge with us. Etienne always makes me think in ways that I haven't had to think before. So thank you for that. So as I said, this is was the last in our series of credibility in neuroimaging seminars. The other two are online, and together, the three of them, I think, are going to be a really nice resource. So I encourage you to share this resource with your students or anyone you think would benefit from it. And also just wanted to plug the BNA Festival of Neuroscience, which is happening in April. And credibility is going to feature very heavily in that. So the plenary session, it's on credibility, and Russ Poldrack, whose name you will have seen all over Etienne's slides and others in the series. Russ Poldrack will be speaking there as well. So we will sign off there. Thank you, everyone, for joining us. Thank you to BNA for hosting the series, and thank you very much for Verena for putting this all together with us. It's been a lot of fun, and we're very grateful to be able to share this with you. There we go. Thank you very much, everyone.
See you soon.